Hello! Here is a quick little video highlighting some of the topics from chapter 14, which is all about cellular movement. So the first set of movement proteins we come across are kinesins and dyneins. And these motor proteins move along the microtubules. So you can see the microtubules in gray, and dynein is shown in yellow and kinesin in red. So what I want you to see from this image is that they move in opposite directions. So dynein is going to move retrograde. And remember, retrograde is toward the nucleus, away from the plasma membrane. And in this case, it's really telling you it's moving towards the minus end of the microtubule, towards that centrosome. Kinesin moves just opposite. So kinesin is moving this way. There's a little kinesin. Kinesin is moving anterograde. Which you will remember means towards the plasma membrane. So this is the outside of your cell. It's also moving towards the plus end of the microtubule. Let me make sure this is not... Yeah, the other thing I want you to make sure you understand is both of these motor proteins require ATP. We'll look at this a little bit more, but the key take-home is they require ATP. So you're taking chemical energy and you're converting it to mechanical energy. So you're converting it to motion. Well, let's look at these two proteins a bit more. Here, what I've circled is kinesin. Right? So you're going to want to recognize that this has this light chain, heavy chain, kind of stick figure structure. And on one end, it carries or it attaches to the cargo. And we'll talk about what that cargo can be in a minute. And on the other end, it connects with or interacts with the microtubule. And again, it's moving towards the plus end, which is anterograde movement. And this here is emphasizing that ATP is required. So in order for these little feet to walk along the microtubule, ATP hydrolysis has to happen. And the cargo, primarily we think about the cargo as transport vesicles. So we've talked a lot about proteins being made in the ER and transported to the Golgi. So that would happen using kinesin. Kinesin can also move organelles. And I should mention, this is also true for dynein. So the cargo can be the same for either motor protein. It's just the difference in direction. Kinesin's really important in building the Golgi. So moving membranes from the ER to forming the Golgi apparatus, because remember that when things are moving from the Golgi, say, to the plasma membrane, they're taking away membranes through the vesicle formation. So if you go back and look at the endomembrane system and all the movement of those little bubbles, those little vesicles, that's all membrane being lost and added to different organelles. And so to keep an organelle intact, you may have to give it more membrane, or you may have to take away more membrane. And the direction of the movement is going to be via either kinesin or dynein. So dynein is our other motor protein. Slightly different structure, and I'll show you one that looks more like the structure from your book in a minute. But what I like about this image is it clearly shows this helper dynactin. And dynactin is what binds the cargo. So dynein interacts with the microtubule, moves retrograde toward the minus end, dynactin moves with it, and up here would be your cargo. A vesicle, membrane from the ER, or I guess from the Golgi, moving organelles around the cell. And if you look back, whoops, you can see that these guys also move RNP, RNA protein complexes. They move cytoskeleton components. They're going to interact with proteins. So remember, the microtubules are the train tracks in the cell, and now we have the trains, kinesin and dynein. Here's a picture that looks more like the classical diagram in your book with this hexagonal shape. This stem is actually the dynactin, and it kind of comes here, that interacts. Let me back up a second. Let me just make sure I have that correct. Just a second. Stalk, stem, lever. Yeah. Connection to cargo. Oh, yeah. Okay, so this is the dynactin. Just wanted to not mislead you. Okay. Again, to make this rotational movement, so it's different than the little footstep walking of kinesin, it's more this rotational power stroke. Also requires ATP. I want you to remember that there's two types of dynein. There's cytoplasmic dynein, which is going to walk those microtubules, and there's accinemal dynein that is important in the flagella and the cilia. So we'll look at that in a minute. All right, so this is just to remind you, okay, you can look at these structures, you can look at something that shows dynactin. I'll always show that. If I ask you to recognize structures, you should understand the general structure of kinesin and the general structure of cytoplasmic dynein, and know that they go in opposite directions. And this is just another image to remind you of microtubules and the endomembrane system. Right? So moving towards the centrosome, the negative end, moving towards the plasma membrane, the positive end, retrograde, anterograde. And now we know where all these vesicles are. We could draw the little motor proteins. Another kind of movement that's not happening within the cell, but it's happening outside, is the use of cilia and flagella. And we are only focusing on, I can't spell, eukaryote or eukaryotic flagella. So bacteria, some bacteria, prokaryotes, have flagella, but they have a different structure, although their function is the same for movement. So flagella are generally longer and fewer. Okay, so this is a typical sperm. There are some protozoa that also have flagella, like Euglena. Cilia are shorter and usually found covering the entire surface of the single-celled organisms, such as a protozoa, such as a paramecium. But then you also have cilia lining your respiratory tract in order to help get things out of your lungs, help you cough. besides being different in size and different in numbers, their movement function is a little different. Flagella are used to move the cell. Cilia are actually used to move the environment, which ends up moving the cell. So flagella move by a undulating, I like to think of it as a dolphin movement. The cilia beat like oars, and as they push the liquid past them, they're pushing the cell, or in your trachea, they're pushing mucus or other things out. Okay, so this week talk about more wave-like, or like, oh, I'm really having trouble spelling tonight, like an oar, and this is more undulating like a dolphin. Well, dolphin. Okay, my computer is not happy tonight. All right, the structure, the reason we're talking about cilia and flagella is that they are made of microtubule bundles, right? That's all the green on this slide. And what's really cool is cilia and flagella are actually wrapped in the plasma membrane. So some books call them internal organelles, but they're also moving stuff externally, so they're not moving stuff inside the cell, say. And where these come from is they come from something called a basal body. And last chapter we talked about microtubule organizing centers, and basal bodies were one of those. And actually what happens is a centriole will be formed, and the centriole will come up to the plasma membrane and kind of turn in, or now we call it a basal body. And basal bodies, like centrioles, have a 13 triplet structure. The axoneme region, so this long part of your cilia or flagella, has what we call a 9+2. So it has 9 doublets and 2 single microtubules. And somewhere in here is called the transition zone, and I don't think they really exactly know how we go from 3 to 2 or 13 triplets to 9+2, but it happens. And the other important set of proteins you can see here are these little orange with blue knobs on them, and that's dynein. So this is axonemal dynein. It is interacting with the microtubule, but it's not really moving along it. It's anchored to it, and it's going to interact and move the microtubules with respect to each other. So this slide is trying to show you that. So here is our 2 microtubule doublets. So say we're just focusing right here. And you see dynein, little guy between them. And as the dynein hydrolyzes ATP and moves, the filaments slide with respect to each other. But there's a very important protein called nexin, and the nexin actually is holding these doublets together. So we don't want just to slide back and forth because you couldn't get motion. We actually want a bend. So the nexin prevents too much sliding, and so you only have a little bit of movement back and forth, and what ends up is you bend. And I like this image here because here, if we're zooming in on a flagella, and remember this works for cilia or flagella in eukaryotes, it's showing you here's the bend, here's the bend. So you can have bend, 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 and then as the bends change, you can start to imagine how the sperm is propelled Another motor protein is myosin. And myosin also has a motor domain, which means it requires ATP for movement. Myosin interacts with actin. And remember that actin is a microfilament, not a microtubule. We are going to focus on myosin type 2. Myosin type 2 is found in muscle fibers. It is also found in the contractile ring. So when cells are undergoing cell division, right, we form this contractile ring that helps break the cells into two. So actin-myosin movement in skeletal muscle and in cardiac muscle, I want you to watch the videos I've put links to on the chapter objective page. I can't draw it, so it's better to watch some animations. The important part, again, you have ATP hydrolysis, which allows the protein to change shape. And you have the myosin head, which is doing the ATP hydrolysis, and you have this cocking, binding, kind of pushing and release. And so the thick and thin filaments of a muscle are going to slide with respect to one another in contraction and then slide back when your muscles relax. So these videos are really good at explaining the motion of actin-myosin sliding filament model and explaining some of the cell signaling. And we're just touching on cell signaling in this chapter, but when we get into Unit 4, we're going to talk a lot about it, and we're going to come back to muscle movement. We have different types of muscles in our body. We have skeletal muscle, which we just talked about, and cardiac muscle, and these are similar in structure. They are different in their signaling. Smooth muscle is very different in its structure. Okay? And we will talk about those in just a minute. So skeletal muscle, go watch the videos. Cardiac muscle, the main thing I want you to know about cardiac muscle is that they communicate, these muscle cells communicate through this thing called an intercalated disc. And in that intercalated disc, if you zoom in here, you see something called a gap junction. We haven't covered these. They are, I think, coming up in the next, coming up in the next chapter. Okay, but gap junctions is for cell-to-cell communication. As you're learning about muscles and muscle contraction and the regulation of it, you're going to see that calcium is a key player. So in skeletal muscle contraction, each of these cells are getting information to contract. In cardiac muscle, the way it works is the calcium goes from muscle cell to muscle cell and gives you kind of this wave of muscle contraction. So think of a beating heart. That whole heart muscle is not contracting at once, but it's actually a smooth little wave of contraction. And again, we're going to get to the details of gap junctions soon, next chapter, but I want you to know that it has a different way of regulating its contraction, and that's through these intercalated discs and these gap junctions. Smooth muscle, totally different structure. It also contracts and it also responds to calcium. The signaling comes a little bit different, and this is showing you vesicles with neurotransmitters. So we're going to get into cell signaling in the Unit 4, but these guys, all these little kind of orange blobs, are going to release the neurotransmitters just like you're learning about with skeletal muscle. You're going to get an influx of calcium, and you then, with these dense bodies, you get this whole contraction of these cells. So it's very different than the sliding filament model. What I want you to understand about smooth muscle is it's still a myosin-actin interaction. But in smooth muscle, the myosin, shown here, and I think this is just so cool that we can see individual proteins or these protein complexes. In smooth muscle, the myosin is kind of all crinkled up here, or what's the word I'm looking for? Can't think of. Okay, and it's not phosphorylated. So hopefully you remember phosphorylation is adding a phosphate group. I draw it as a plus P. Okay. When ATP is hydrolyzed, we lose a phosphate group. Okay, so phosphate groups are super important in cells. When the myosin chain is phosphorylated, it straightens out, and now it can interact with actin. The key here, you'll see in your learning objectives, is MLCK, myosin light chain kinase. Kinase is a protein that adds phosphate groups. This kinase is under control of a protein called calmodulin. Calmodulin is under control of calcium. So all of our muscle contractions are regulated by calcium. The signalings come in slightly different ways. This case, the calcium is telling calmodulin to activate myosin light chain kinase to add phosphates to myosin light chain to allow the myosin light chain to interact with actin and, contract your smooth muscle. Okay. Okay. Cell crawling is also talked about in this chapter. And cell crawling or cell movement like this, we will come back to when we talk about cancer cells and metastasis. What I want you to make sure you understand is this cell movement is all due to actin. Okay, so your microfilament, and you get polymerization and breakdown or depolymerization. And that allows a push-pull, so the cell can push out that cytoplasm, that cell membrane, into what we call this protrusion. Adhesion points, we're going to talk about this again, chapter 15, next chapter. It lets go, and now that this is broken down, it can kind of move forward. So our actin microfilaments are important for cell movement. Again, this is something that is much better in a video. Okay. So this video is an animation about cell crawling and how a cell moves, and it goes through lots of those terms in your objective. And these videos are just fun 30, 40-second videos to actually watch cells moving under the microscope. Chemotaxis is another concept we're introducing in this chapter. Chemotaxis means that there are chemicals that cells respond to. So this is showing a chemoattractant, right? A chemical that attracts cells. This is super important in your immune system. So this is a neutrophil. It's being attracted at this site of injury, and it's going to move. And how it's moving, it's not showing it great here. Some roll and tumble, some cells crawl, but what's important is it keeps having these contact interactions, and then that neutrophil is going to slide right in between those endothelial cells and go help repair your tissue. Again, some cool videos just showing cells moving towards chemoattractants. Concept to understand. We will come back to this more in Unit 4. And finally, your last couple of objectives ask you to understand amoeboid movement and how that is different than cell crawling, and cytoplasmic streaming. And I think you really need to see it to understand it. So these two videos show you amoebas moving, and this video, shows you cytoplasmic streaming in plant cells. And hopefully in your general biology class you got to watch this in person. All right, that is it for chapter 14. Lots of videos by Dr. Duncan if you want to go through things in more detail. And remember, there are some molecular mechanisms of muscle contraction and a little bit about signaling posted on the objectives and videos page. Thanks.